So this is the chapter 2 study points. So basically we need to know, this chapter is all about like microscopes and staining. So what we need to know about compound light microscopes is basically it was invented by Hooke and has a magnification up to 1000x. Total magnification is what's it called? It is calculated by lens times objective. So for example, if 10x lens, we're using a 10x lens and then we have it on a 100x objective, you multiply that together and you'll get a total of 1000x magnification. A question will come up on that exam. So why is a light microscope referred to as bright field? It's referred to bright field because it's basically replacing a dark image, which is our microscope slide, and is being illuminated by a bright background. And why are microbial cells stained? So basically, dyes are used to increase contrast and provides information on cell morphology. So basically, like, shape and just other information that I'll get into later. And explain how bacteria are stained. What are the critical parts of staining? So basically, bacteria are either heat fixed or chemically fixed onto a slide and then dyed with either basic or acidic slides or acidic dyes, sorry. And what are basic and acidic dyes? Explain how they work. So basically, basic dyes have a positive charge and stains cells. Acidic has a negative charge and stains the background. What are the reagents used in gram stained? So the mnemonic I use is VIDS. So violet, iodine, decolorizer, and saffron. So crystal violet basically stains the cells purple. The iodine basically is a mordant that makes the wall a little bit like looser, if that makes sense. So that when we decolorize it with alcohol, basically it washes away the purple, either purple dye from gram-negative cell walls, or it remains purple in gram-positive cell walls. And saffron is the final step and basically counterstains. It makes the gram-negative bacteria either pink, or if it's a gram-positive, it remains purple. So basically, the reason why we're doing gram staining is to distinguish whether or not like bacteria is either gram positive or gram negative. So gram positive bacteria basically has a thick layer of peptidoglycan. That's what you need to know. It has a thick peptidoglycan and basically retains stains better than gram negative bacteria, because since it's gram negative, it has a thin layer of peptidoglycan. You need to know that. So since it has a thin layer, it does not retain stains as well as gram positive. Hence why it's pink and why the purple dye is washed away in the decolorizing step. And the most critical step in staining is the decolorizing step, because if you do it for too long or for too short, you can get inaccurate results. So for example, right, if you're using too little alcohol when decolorizing your bacteria on a gram-negative bacteria, you could get a purple result, even though it's supposed to be a pink result since it's a gram-negative bacteria. Just basically means that you didn't wash away the purple dye enough, And then you need to know about bacteria and which do not stain, and which do not stain in the gram stain and why. So acid fast do not gram stain is not it's not good against acid fast bacteria, because the difference between like a gram positive bacteria that is not acid fast and one that is that acid fast bacteria has a thin layer of mycolic acid on the cell, outer wall, right? So basically it's like kind of like a shield that prevents purple dye from penetrating and targeting the peptidoglycan layer, right? So acid fast bacteria need acid fast stain test because crystal violet dyes cannot penetrate mycolic acid cell wall layer. Mycoplasma also lacks a cell wall, so you cannot stain mycoplasma bacteria due to the lack of peptidoglycan, because that's the whole point of gram staining, is to target that peptidoglycan layer, right? Distinguish between simple staining and differential staining techniques. So simple staining is your basic and acidic dyes. And basically all the cells appear the same color, while differential, you have to think about our gram stain or acid fast stain. Basically uses multiple stains, right? So you're using the saffronin and the crystal violet. While special stains like flagella staining, endospore staining, and capsule staining basically are used to identify whether or not that bacteria has that structure. And I just went over that. What is the clinical relevance of acid fast stain? So patient with Mycoplasma pneumoniae cannot have isolate bacteria stain via gram staining. Patient with Mycoplasma pneumoniae cannot have isolate bacteria stained with gram staining because what's it called? They have that thick layer of mycolic acid, right? So we cannot stain Mycoplasma pneumoniae with just normal gram stain. We need the acid fast stain to penetrate through that mycolic acid layer. In microscopy, what is resolution? How is it improved at high power? What oil is used? So resolution is basically how clear we can see a bacteria using the microscope, and it's improved at high power when we are on like 400x, right? With oil to prevent light refraction. What are the smallest objects that can be visualized with a light microscope? It will be bacteria and some large viruses, but what you need to know is bacteria is the smallest. Case study: how did the microscopy, that's how I pronounce the word, gram stain help the clinician determine the causative agent of the patient's wound infection? So basically, the patient had this sickness, unknown sickness, so they isolated that bacteria, and we looked under a microscope with the bacteria was stained with gram stain first off. Then we took it under a microscope and we examined that it was spherical and in clusters. So we basically were allowed to examine the cell morphology, and then we also noticed that it was purple, which means what? It's gram-positive bacteria. And both of these characteristics are similar to those of Staphylococcus, and I think it was like Staphylococcus aureus. So that's how we can use gram staining and microscopy to understand what we're looking at. Good luck.